Welcome. Welcome to the workshop, Clinical Research from Study Design to Scientific Presentation. I'm Aditya Bardia, breast medical oncologist at Massachusetts General Hospital, Harvard Medical School, Boston. This workshop is focused on clinical research. We have 4 topics: selecting rational biomarkers for clinical and translational studies, how to design an efficient clinical study, how to include patient advocates in research, and finally, how to present study results effectively. At the end of each presentation, there will be 5 minutes for live Q&A. You can use the web portal to send in your questions. So without further ado, it's my pleasure to invite Dr. Justin Balko from the Vanderbilt University Medical Center, who will be reviewing selecting rational biomarkers for clinical and translational studies.
Thank you very much. It's my pleasure to be here in person for the first time in 2 years, and so it's fantastic to see all of you, or many of you again, who I've met, and those of you who I've not met. I am a professor for cancer research and co-leader of our breast program at Nashville, Tennessee at the Vanderbilt Ingram Cancer Center. So I'm going to be talking about rational biomarkers in clinical and translational studies, just a few pieces of advice from my own experience. These are my disclosures. We get research funding support from Insight and Genentech, and we have several patents pending for biomarkers in cancer therapy. Our objectives today are really to start simply and just align the basics of what encompasses a biomarker, how a solid biomarker plan or an intact biomarker plan can influence the outcomes, the productivity, and the impact of a clinical trial, and then some logistics and considerations in design. And then we'll move on to briefly review some of the novel technology platforms, in particular those that I think are more amenable to small phase and to some larger phase clinical trials, rather than going extremely deeply into emerging technologies. We'll talk a little bit about how they work and present some examples and some of the pros and cons of using each one. So just to start off very simply, what is a biomarker? The NIH NCI defines a biomarker as an indicator of either early disease detection, so for instance, a diagnostic biomarker, those that can predict or identify disease progression and outcome, and those would be prognostic. And then finally, an example of one that's truly predictive, meaning that it differentiates between a response to a particular type of therapy. We use these quite frequently in clinical practice in breast cancer. So here's just some examples with different tissue sources or different technologies. So for instance, immunohistochemistry, which is one of the older technologies we've been using for quite some time. Examples would be estrogen receptor, progesterone receptor, HER2 positivity, Ki67, PD-L1, and TILs. As far as RNA, so a different type of analyte, we know these to exist in the form of typically like Oncotype DX assay, the MammaPrint assay, and also the Prosigna assay. DNA can also be used as an analyte. This is quite popular. We've been hearing about NGS studies. We can look at tumor mutation burden as an FDA approval for the addition of IO therapy, or the treatment with IO therapy, rather. And you can see I've listed several mutations here, or genes in which mutations can predict a response to therapy. We don't have to just think about biological analytes, so RNA, DNA, protein, but we can also think about imaging. And so there's a number of different types of imaging right now that are either been routinely used in clinical practice or in clinical trials. And then, of course, there's others, so things like we would typically get in clinical labs, like ANC, and utility of growth hormones. So what's the purpose of a biomarker plan, particularly in thinking about early phase clinical trials and some later phase clinical trials? So certainly there's an aspect of clinical development. So in an early phase clinical trial with a new agent, we might be thinking about using a biomarker as an evidence of, or evidence for pharmacodynamic activity of a drug. For instance, inhibition of ERK or an element of the MAP kinase pathway, showing that our MEK inhibitor is working in the tumor microenvironment, or even sometimes in peripheral blood samples. correlates. We can use it to help prognosticate patients, so perhaps to identify a very high-risk group of patients to which we can make our effect size and the clinical trial design more robust. We can also use it eventually for prediction in patients to try to identify a subpopulation of patients that might need to be treated with that agent in order to derive a specific pathway benefit. We can also use this sort of back in a translational appeal or a translational approach back to the lab, so it can inform on biology. So retrospectively, we might use a biomarker plan or correlatives from tissue-based trials or tissue-based samples to try to understand mechanism of resistance or sensitivity. And we can use this to guide bench to bedside or bedside to bench translation. It can also tell us a little bit about the pharmacokinetics and pharmacodynamics of the drug and how our future trials might need to be adapted. So the NCI has 3 formal classifications of biomarkers in NCI trials. These start at the most critical, which is integral. So these are used for patient selection. They have to be performed in a CLIA lab, and they may require an IDE. They're typically, one defines as an integral biomarker as you need to be able to perform that test conclusively in order to conduct the study. The next level is integrated. So this is typically used for patient description or stratification of the final results. It still has to be a robust assay, and hopefully, ideally, that would be a CLIA-ready assay, but it does not require an IDE. And then finally, the majority of early phase clinical trials have exploratory analyses. These are often more multi-analyte or multi-data point types of analyses that really help us tell across the entire population what might be going on with this new drug or what might be going on in certain patient populations that might be associated with sensitivity or resistance, where we really don't have a good hypothesis yet. And so we really want to prioritize in clinical trials, particularly larger clinical trials, and integrate an integral or integrated biomarkers very early in development, because it can actually be quite difficult once you get to the phase 3 trial if you haven't included these types of biomarkers in the phase 1 and phase 2 studies. It can be kind of difficult to get these ingrained into a good phase 3 trial. So what are the outputs of a well-designed plan? So of course there's scientific discoveries, right? This is what we all want to make. We all want to inform on the next clinical trial or what we should be doing in the lab in order to make treatments better. These, of course, are integrated also with grant funding, which is something we all need to stay alive and to continue doing the research that ultimately results in high-impact papers. And then finally, the whole reason that this whole cascade exists and why we're doing this is to ultimately impact patients and to impact the way that we treat patients. So let's talk a little bit about logistics and design and some important considerations before we figure out exactly what we want to do as far as the biomarker or correlative plan. So I think it's important to think about balancing expectations. As a postdoc or a research assistant professor, when I was just sort of starting my lab in the early years, I would be conducting or participating in an early phase clinical trial in breast cancer patients along with my clinical colleagues, and had all these great designs about using these really high-level technologies to figure out what was going on in these samples, because it was always so difficult and so precious to be able to get these clinical samples from patients that were systematically treated on clinical trials. But we really have to take a step back and think about what is financially feasible. So many of these very high-level technologies cost a lot of money, but they do provide more data points. So they kind of give us more shots on goal as far as discovering a little bit of biology from those samples. In many cases, my advice would be to consider partnering with that developer. The reason for this is these emerging, very high-level technologies often aren't very well validated yet. There's a lot of excitement around them, but they can be much more complicated than we all think they will be to actually analyze the data or even conduct that type of technology, even though you may have equipment to do that in-house. And so I would consider trying to contact the developer and partnering with them. Sometimes you can get funding to do these, but you'll also get expert analyses from the developer that's actually very engaged and has a lot on the table as far as they want to make this work. What is statistically feasible is another big question. So, in a small phase 1 single-arm trial, one has to think of how much do you want to invest in these samples for a very high-level technology where you may not really be able to or be predicted to be able to show anything that is actually statistically sound or isn't just due to interpatient heterogeneity given the small numbers. So it's important to balance that expectation. It's also important when you're designing it, but before before the clinical trial is conducted to consider that you're going to have some level of sample attrition. And finally, if you do all these different studies, will you still learn something? So one exercise I always like to do when I'm thinking about employing a new technology on clinical samples or designing a correlative plan is I like to write out some potential hypotheses. Maybe I don't have all of the data to support those hypotheses, but I write out specific hypotheses that patients with a higher MEK activation transcriptional score will not respond to this therapy. And if you write all those out, you really have some guidelines on what are the most important questions here and what can we do in the most financially and statistically reasonable way where we still have some pre-identified questions. What's logistically feasible? So this is something that I've learned many times in the course of being a translational scientist. That's that the lab work, too many things can result in everything falling apart. I can't tell you how many times one of my postdocs or my graduate students has thought about, this big exorbitant mouse study that they want to do, and all these different sort of ways we're going to do flow cytometry, we're going to do this, and that. And luckily I have a very cohesive and cooperative lab that will really invest everything in their day's work, all to help out one of the graduate students or postdocs who conducted a study. But probably, if you've worked in a lab before, you know that trying to do too much can cause everything to fall apart. And so this is definitely true in designing correlatives, particularly when it's not just derived from some kind of typical analyte like a formalin-fixed paraffin-embedded sample, which we can do. It's safe, it's stable, and then all of our assays are downstream of that. So something that might require a lot of, at the point of collection, a lot of prep work and a lot of logistics. You have to think about, for multi-center trials, is it necessary and do you need to ship samples immediately to a central lab in order to minimize heterogeneity, or even to have the equipment to actually process the sample? And so, things like cell-free DNA, where now we have Streck tubes that keep the samples stable for a couple days, or you can freeze them. Do you have all the equipment at all of your sites to be able to do that and get it to a stable position? Or is it going to really increase the logistics or the logistical strain on the clinical trial? And of course, that's less of a problem with single site trials. But often, the more sample types that you try to collect, the more logistics that are engaged there. So this is just an example of a tissue and blood study that we were looking at doing through one of the research consortiums that I'm a part of, the Translational Breast Cancer Research Consortium. And it's not really important to read everything here, but it took a lot of thought on how are we going to maximize a couple of tubes of blood from a patient at different time points and try to really streamline the maximum we could do, pretending that this was such a limited resource, and granted, peripheral blood is not quite as limited as tumor tissue, but it is still limiting, and it's still, we don't want to be wasteful in ways like this. So we thought about all the different types of analytes, whether that would be plasma for cell-free DNA analysis, whether that would be PBMCs for flow cytometry or CyTOF analysis, whether that would be DNA for controlling for whole exome sequencing, so we needed germline DNA, or possibly in this particular study, we were thinking about HLA types, we were thinking about a number, T-cell receptor sequencing in the peripheral blood. And so what we really did was we thought about and we tested out with all the different types of tubes that we could collect with minimal volumes, what are the ways that we could sort of layer on or multiplex sample collection into different types of analyses. And in designing things like this, don't just think in the moment of what you wish you had right now, but imagine what you might wish you had when that trial is actually done, or even a couple years down the road when you might have bank samples. What might be important? What can you foresee? Sort of get out of your crystal ball and think of what might be useful from this clinical trial in the future. So now I'm going to spend the last 4 or 5 minutes on platforms. So these are a few that we've become really familiar with in my lab and started to use. Some of them are tissue platforms that we've had access to, that we've tried, we've steered away from for cost purposes. But I'm going to give you a brief overview of several different platforms that I think are important to talk about. So we usually think about technology platforms for correlative analyses in 3 different ways. So bulk, this is where you're taking a section of tissue or a core biopsy, and you're putting it in some kind of analysis pipeline that's essentially going to homogenize the entire sample. You're getting everything that's in that, for instance, core biopsy or a peripheral blood sample, and you're going to look at things. So there's advantages and disadvantages to this. if you think about this, in bulk, you're getting a net conglomerate of everything that's in that sample, and it's easy. Logistically, it's quite easy. You don't have to do any kind of separation, but you're also losing what's going on in a different cell population or a single cell level, and also spatial analysis. So in situ technologies, like such that we would do from formalin-fixed tissue, these are actually quite informative because they conserve the spatial profiles of the tumor. So they tell us when are T cells interacting with the tumor cell, how is the stroma or the vasculature associated with different regions of the tumor. And there's a lot of advantages. You'll hear a lot in the last few years, and also even at this meeting, on spatial analyses and what kind of additional insights they bring to the table. And then finally, single-cell dissociated. So this might seem like the best, single-cell RNA-seq. We get these beautiful plots. We know what's going on to some degree in all cells within our sample. This has both pros and cons. Yes, those are true. Some of the negatives, for instance, from single-cell RNA sequencing is you're missing a lot of things. You're only getting the top 2,000 to 5,000 transcripts that are expressed in each cell. You're also completely losing the spatial analysis. So you have no idea where those cells came from. And then finally, the logistics associated with getting most solid tumors into a single-cell RNA-seq pipeline can be quite laborious. They can be difficult. They can end up in a low success rate, or at least a significant sample attrition. And so one needs to balance what one will learn by employing these technologies, and as well as the cost versus what's to be gained. So just starting off with some of the bulk, this is probably the workhorse right now of most clinical correlative trials, right? Doing bulk RNA analysis. This gives us, it leverages huge databases of gene expression signatures, of knowledge bases from many clinical trials. And so I would support almost every clinical trial where you're collecting a tumor biopsy to conserve some of that tissue for bulk RNA sequencing. Not only is it informative to yourself, it's informative to the clinical trial, but ultimately once you publish those results, it's going to be informative to the rest of the breast cancer and cancer research community. So typically, there's a number of different technologies here. I think the two most applicable are RNA sequencing and nanostring. And I've listed some of the pros and cons here. I'm not going to read everything. The biggest deal to me is the speed, the time to analysis, and the ease of analysis with nanostring. Their typical N-counter analysis. Versus RNA-seq. So the pros and cons here are that nanostring is incredibly useful for formalin-fixed paraffin-embedded tissue. It's nearly as good as fresh frozen. Now, you don't get quite that same success rate with RNA sequencing, but it's improving. However, the cost of RNA sequencing is rapidly going down, and you get more data points from RNA sequencing, whereas the price of running a nanostring panel has remained fairly stable over the last 5 to 10 years. So now moving on to something else that I think is incredibly useful in most tissue-derived correlative plans, and that's in situ analyses. So I'm starting off with multiplex immunofluorescence. We're doing this for many years, but the technology in the field is still making huge strides over the last 5 or so years. So this is relatively simple. So if we just think about a simple 3 to 4 color assay, it provides single cell level data. It still requires a lot of validation. I think this is one of the things that the more in situ technologies that we see in the field, the more that become available to us at different institutions, we still have to remember the amount of validation that requires in truly knowing that the answer we got is actually what's there in the tissue. There are improvements in automated staining protocols and analysis workflows. However, it's also important to remember that typically this is limited to 3 to 4 plex, meaning 3 or 4 analytes in a sample. Now, there are certainly technologies that go on there, up to 9 plex, for instance, with the Opal TSA approaches that's owned by PerkinElmer and now to some degree Akoya. However, it's important to remember also that going up to a custom 8 or 9 plex assay requires an intense amount of validation. It can cost several $100,000 just to develop an assay, and it can cost 6 months or a year to develop that assay. So those are important considerations. It's also excellent for large-scale analysis if you are able to get that 3 to 4 plex assay up and running. Now I'm going to talk a little bit about something I think a lot of us are starting to become more familiar with, geomics, which is from nanostring. So the workflow here is essentially what you're doing is you're taking 30 or 40 different monoclonal antibodies with their protein workflow, and you're conjugating oligonucleotides to those that are attached by a photocleavable linker. And so what this does is on the slide, you can't see anything from those markers, but you also co-stain with a couple of typical immunofluorescence markers. You then image that slide according to those those fluorescent markers and select different regions or different cell populations based on your architecture markers to hit with a UV light. Once that UV light hits, those oligonucleotides are cleaved off. The machine sips up those oligonucleotides, then counts them on a machine. So essentially what you're doing is you're getting regional analyses of 30 or 40 different markers, sometimes in particular cell populations, like only the pan-CK positive tumor cells, or T cells, for instance, or you're looking at a certain region, what's happening at the tumor margin, what's happening in the core of the tumor. This is really useful for capturing cell populations, but there are limitations to think about. This is not a cheap technology, and if you're using things like core biopsies, one has to ask, are you actually getting the most out of your money here? Because you're only going to be able to select several regions of interest for the minimal size of those regions of interest on a small biopsy. This is just some example of some data that we generated. I think this is actually one of the better uses of the technology, which is to mask your sample based on those immunofluorescence markers that you've used. So what are all the things that are going on in the CD8 positive T cells within a certain microenvironment or within a certain tumor? What are the monoclonal antibodies or the signals? What are what's going on in the tumor cells? And then to compare those. So again, we're not getting single cell level data, but we are getting cell population level data. The next one is AltiView. So this is a barcode PCR technology. So these, I like to call them canned 5-plex assays. So you can develop your own assays through AltiView, but the real strength here is that they have pretty standard and pretty interesting 5-plex panels that you can just order off the shelf, are highly validated. And if you have the right equipment, as many institutions do, you'll be able to look at these without any additional validation. You should certainly test them out in your hands. But I think that they offer a lot of sort of plug and play technology. However, they are extremely costly, as I think many of you guys know. And so this is one of the reasons why it's great to partner with industry when thinking about employing some of these technologies in larger numbers of samples. And then finally, Akoya Codec. So this is a 40-plex analysis with a strip and reprobe. So essentially what you're doing is you're using 4 to 5 different channels. You're binding all the antibodies, you're stripping those off, and to be quite basic about it, and then reprobing with a new 4 or 5 set. You're imaging in between each one of those. The equipment for this is surprisingly cheap. It's automated, but the optimization and validation is key. So this is one of those things where simply because you can do 40-plex and you can get an answer, you have to remember that validation, not only of the panel in your own hands, but also of whatever you find in that analysis, you should probably do by a more low-plex technology. And so there are some others. There's Recore Cartana, which gives a subcellular localization. There's Visium, which is a full transcriptome in situ across the entire specimen. These are earlier technologies. They're in many cases quite costly. They're probably not amenable to large numbers of samples, but they can tell you where to look in the rest of your samples. So that's a good point too, is if you want to employ one of these higher level technologies, to just use it on a few samples and then figure out what your exploratory marker is that you want to test across the rest of your samples. They all have their own positives and negatives, which I've listed here. And so in conclusion, our biomarker correlative plans can exponentially increase the amount of information that we learn from even small clinical trials, but we really should deploy these in a thoughtful manner. You don't just want to do everything under the sun because you have the tissue. You want to ask thoughtful and intelligent questions, and also those that can actually impact future studies with those agents or in that particular indication. Any exploratory analysis you have can pose a significant risk. We don't want to chase unicorn zebras just because we find one thing out of 10,000 of different data points that we looked at in a clinical trial. This could actually, maybe the true danger in this is spending months and months into years of sort of following that up with no validation that it's actually not just a random finding. And of course, we always want to keep abreast of new technologies, but also take the time to understand the limitations and the caveats. And with that, our acknowledgments of some of the funding sources my laboratory receives, and this picture taken recently in my lab at Vanderbilt on a beautiful day. Thank you, guys.